Hi, this is Sadashiva Pai from Science Mission. So today we have Dr. Joseph Dougherty, Associate Professor, Department of Genetics and Department of Psychiatry with us. So he's from Washington University School of Medicine, St. Louis. So Joe has won several awards. To his credit, he won Harvard Hughes Medical Institute Pre-Doctoral Fellowship, K99, ROO, Serotin Autism, and Investigating Cell Types for CNS Disorders. So Route 28 Summit in Neurobiology Award for Excellence. He was highlighted one of the genome technologies up and coming investigator in 2012. So in addition, he also has won several grants and has published many papers in high impact peer reviewed journals. So welcome, Joe,
Hello,
to
thank you.
Science Hangout.
Yeah.
Can you please talk about yourself, your background, education, give an update on like what you are, like what you do, kind of thing.
Sure, yeah. So I've had the lab here at WashU for
Mhm.
8 years now. So I'm
Okay.
used to describing myself as a new professor, but I guess that's not true so much anymore. Um,
Okay.
my original interest in science actually came in through psychology as an undergraduate, sort of biological psychology from a small
Uh-huh.
undergraduate institution in Missouri called
Uh-huh.
Truman State. And I was very interested in I think what drew me into science was sort of an interest in how the mind worked, but I wanted to be able to do methods that were physiological and you could sort of test rigorous questions. So for graduate work, I went and did molecular neuroscience with Dan Geschwind at UCLA in the early days of
Uh-huh.
microarrays and high throughput transcriptomics. And I worked there on a lot of defining differential gene expression in neural stem cells and genes that regulated proliferation versus differentiation, which I think was
Mhm.
interesting and a great experience. But by the end of it, I realized I wanted to move back up towards questions of things that were involved in the mind. So for a postdoc, I went and worked with Nat Heinz at the Rockefeller University. And I was
Mhm.
really excited about that because it gave me the opportunity to take kind of these skills I built in transcriptomics and genomics, but actually take them up into a functioning brain because Nat's lab at the time was developing these methods for doing cell type specific in vivo profiling. I'll actually mention some of those in the talk as the starting, a little bit of the starting point. And excuse me for coughing. I've got kind of a bad cold. I've got some tea going, so hopefully
That's
I'll
okay.
be able to keep it restrained. Um, so the mouse was sort of the right level organism for me where had the control to be able to do well designed experiments of cause and effect in terms of how genes regulate behavior and how but also um, was kind of a high complicated enough organism you would have things like behavior and cognitive processes going on more so than kind of cell culture work. So that kind of ended up being where I settled. And then for my lab, what we focus on is a mix of different things. So we're trying to understand broadly how genetic mutation changes circuits in the brain in a way that results in psychiatric disease. Our disease focus is mostly autism. We do a mix of mouse modeling of genes that cause autism, focusing on those that come out of high impact studies from collaborators,
Mhm.
especially those things that regulate transcription or translation. And then on the other side of the lab, we do a lot of technical development around understanding how transcription and translation are regulated in the brain, bringing new methods online, um, focusing on cell types we think are important for disease. And so today I'll be talking mostly about some of that technical development work and how it led us to discoveries of local translation and astrocytes and some of the translation regulation work we've been doing
Okay.
recently.
Can you talk a little bit about your lab, your grad students, postdoc? Give a like idea on like how is your lab, like how big, what are
Oh, okay. We're about 15 people right now.
okay.
It's a mix of, let's see, we have two postdocs and an instructor at the moment at kind of the senior level, uh, two technicians and a programmer in terms of staff, and then about half, 7 or 8 graduate students, depending on how you count the ones that I jointly mentor. Um, it's a really good group of people. It's probably, I try and structure the lab so that I get to interact with everybody as much as possible because that's what I enjoy about science is interacting with other
Mhm.
scientists. And I kind of view the people in my lab as my long-term collaborators that I get to work with. So I try and meet with everybody kind of weekly to hear what they're up to. We either do it one-on-one or kind of a mix of one-on-one or small groups who are working on related topics, so that can be good information sharing across teams working on related projects. Yeah, we do a mix of sort of everything from molecular to computational to behavioral work. Most people do kind of more than one thing. So I think that's important to have, especially for training, to have really a multifaceted experience in biology, because it requires expertise in so many different domains. Even if your career is going to take you really largely into one of those, having some exposure during your training to each of them, I think really helps make you a strong collaborator going forward and a well-rounded scientist overall. I think the mission of the lab is really to both produce really good science, but also to really produce very strong scientists. So I'm really kind of focused on providing a good training experience for the students in the lab, trying to teach them how to run a project independently. And I view most of what we do is really, I'm pitching ideas to them, they're pitching ideas to me, and the thing that ends up happening is sort of a hybrid of those. Those ideas coming from them and coming from me.
And regarding the research is a long drawn process. It takes years and all those things to publish a paper. So there is always ups and downs. So how do you keep them motivated? So make sure that they come to the lab and do the things and always in a positive mental state.
I mean, it's hard, right? Sometimes you'll go through periods in graduate school, especially where things just aren't going to work for a while. I think I spent 6 months in graduate school at one point hybridizing what I thought were these custom microarrays with precious RNA that I had to keep. It would take me like 3 weeks or 4 weeks to do this long differentiation protocol to harvest this trace amount of RNA and then label it. And I'd scan this slide, it would be blank. And so I thought I was hybridizing months and months, hybridizing these custom arrays and not getting anything. Until I think finally one day I stain a slide with DAPI and realized that there's absolutely nothing on the slide. And I just spent 6 months trying to hybridize blank slides that I'd gotten from a collaborator. So, I mean, these tragedies in science kind of happen. And I think what has to keep you going, well, first, I just try and hire people that are very excited about doing science to begin with. You have to be internally driven to take on this career, to take on these challenges, and patient and excited about the next experiment. And then I try and always make sure that people have a couple of different things to work on, so that when you hit a wall in one area, you still can have movement in the other. Because I think it's good to have a kind of a balance of projects that are high and low risk, so you have something going on that's positive. And then I guess the third thing is to get time away from the lab too. When things aren't working, it's a good time to go, well, go take a break and just go read for a while, go think for a while. And so let's try something, if it really, once you've tried an experiment 2 or 3 times with minor variations, if it's not going to work that way, it's really, let's try something completely different, come at it from a completely different angle. Hopefully that helps keep people motivated and excited about what they're doing.
Yeah. And regarding your hobbies, like I know you are busy with all the projects, so many people in the lab and all. So how do you balance that work-life thing? Like, what are the other things you do?
Well, I've got 2 elementary school age kids at home, so I think my main hobby is whatever their hobbies are, kind of playing with them. And when I try and to structure my free time, it's around the days that they're off. So yesterday they were out of school for parent-teacher conferences, so I took them skiing at this little local Missouri ski resort for the day. I mean, it's not great skiing in Missouri. It's kind of like skiing on mashed potatoes. It's really slushy, but it was really fun. So I like to do those kind of outdoorsy things with my kids when I have time. I read. I don't know. I have a lot of different interests. I like to try different restaurants in St. Louis. There's a pretty good food scene. Go through music. Yeah. It's good. It's important to take time away from the lab too and recharge.
And regarding mentorship, like that's an important part of the whole research aspect. So how do you view it? Like, can you talk something about this mentorship? People helped you, and how you plan on doing it to your students?
I have a lot of thoughts about how to. I mean, I could give a whole hour. I think about mentorship and training people. It's interesting. Science is an interesting kind of training because I think a lot of students come in kind of focused on the methods and learning methods and techniques, and those are important for sure, but they're just a foundation to what we're really trying to train you in, which is how to move into an area, define a key question, and design the experiments and execute the experiments to address that question. Because the actual methods that you learn, I mean, there's so many things that I can think back on in graduate school that I spent months and months mastering that are now completely obsolete. Like I learned how to build a robot to print little microarrays, and that's not useful anymore. But learning how to read and think about science is a skill that can last forever. So I usually do that through writing a lot with students. I had everybody write grant proposals, like training fellowships as a way of kind of guiding them through thinking about the process of an experiment ahead of time. It's a lot of, a lot of meeting and discussion. I try and prompt them to try and figure out how to get them to think of the next experiment on their own. And by the end, what I'm hoping, early on, often I'm sort of pitching experiments to them early in the graduate career, but by the end of the career, the idea is they're kind of coming into my office excited about the next experiment, what they want to do. And I'm just there sort of like, oh, here, helping them guide their thinking a little bit. Have you thought about this control or that control? Or, but that really trying to teach them how to kind of be reading the literature, thinking about the next experiment on their own, asking for help where you need it and training where you need it. But really, teaching them to be independent is the goal overall.
Okay. Now going back to the research, talk about some of the projects going, and you can start presenting once you are into.
Oh, okay. Well, I already kind of talked about a few of them.
Yeah.
in big picture. I'm excited about everything the lab is doing.
Okay.
Always. We have one set of people working on mutations in mice for genes we think cause disease in humans. A big focus there is the Williams syndrome locus, which is a 28 gene deletion, but one of the dominant hypotheses right now is that it's a couple of transcription factors in there that are mediating the phenotype. And it's an interesting phenotype because you can get changes in social motivation depending on whether you get deletions or duplications in this region. So we're trying to model that in mice and understand at a molecular level what the consequences are and a behavioral level. On the technical development side, I think I'll be talking a lot about that, the translation regulation stuffs now, so preview that. The other kinds of technical development that we're doing is some cell type specific epigenetic profiling methods that we've been developing. There's 2 preprints we just put up on that. It's called the calling cards methodology. So it's a single cell version and a targeted version. So those are up on bioRxiv. My name's on them, but the lead authors are Alex Kamak and Arnav Mugdal. So if you want to look those up and hear more about that project, you should go check out that work. We're also widely collaborative, so there's almost more collaborations that I could ever possibly discuss in this. Since we do a lot of technology development, we're always happy to share whatever we're developing. So there's a lot of collaborations because of that. And then there's this translation kind of regulation methods we've been developing that I'll talk about now.
Go ahead. You can share the.
You put the slides over. Okay, I keep looking over there because that's where they are for me. Let's see. Click the shareable thing.
There you
go. Okay, you guys see the screen now?
Yep.
Do you see my mouse when it's over the screen? Does that work as a little bit?
Yeah, I can see that.
Okay, excellent. So today I'll try and spend the next kind of 30 minutes or 35-ish minutes talking about 3 little stories in the lab that are around new methods we developed to study translation regulation in the brain, how we came to be interested in that, and then what we ended up discovering, some of the things we expected to discover and some of the things that were surprises to us. Is the sound okay too? Can you hear me? Okay. Good. All right. So as I kind of alluded to earlier, I'm interested in how different cell types express different genes in the brain. So as a neuroscientist in training, I'm always required to show at least one image from Cajal. It's in the contract. So here's my Cajal image. This is an image from the cerebellum, and I just put it here to illustrate that even in a relatively simple structure like the cerebellum, you have a variety of a wide variety of morphological diversity and presumably equally diverse functions across these different cell types in the brain. Yet every one of these cell types has an identical copy of the genome. So how do you get this diversity of morphology and function? You do it by choosing what fraction of the genome you express. So every cell type has a unique gene expression profile. At the beginning of my postdoc, I was very interested in understanding which genes were defining what each cell type, particularly for those cell types that might be important for psychiatric disease, as you have thousands of cell types in the brain, but presumably only a subset of those are really driving the circuits that disrupt brain function in psychiatric disorder. So how do you measure gene expression in a complex mix? So this is not actual data. This is just a cartoon, obviously. But the problem when you're trying to analyze a mix of different cell types is that here cartoon is sort of green triangles for neurons, or red for astrocytes and blue circles for dendrites. Is that whenever the most methods to measure gene expression traditionally would be grinding up this whole tissue and measuring the RNA in bulk from it. And you can see sort of that you would end up getting a mix of RNAs from the different cell populations. So how would you measure this in specific cell types? What my postdoctoral lab, particularly Miriam Heyman, who was working with Paul Greengard and Nat Heinz at the time, what they kind of settled on was this idea that if you really want to purify the mRNA from those cell types, you don't have to purify the cell types themselves, which could be challenging in an adult brain. Rather, if you just pull the ribosomes out of those cells, any mRNA that's being translated from in those cells would be captured, and you could then kind of look at the cell type specific gene expression in that way. And so they developed this method called TRAP, translating ribosome affinity purification, wherein you use mouse genetics to drive GFP expression in a specific cell type by using a cell type specific promoter. Here's an example from serotonin neurons we did. And what you drive instead of normal GFP is GFP fused to a ribosomal protein. So on one hand, you get fluorescent labeling that you can see and confirm the expression. But on the other hand, you also get an affinity tag on the ribosomes. And so if you use antibodies to GFP, you can pull the mRNA out of a lysate. Even after lysing an entire mouse brain, you can select for the mRNAs that came from the cell population and enrich for those. And sort of a standard comparison we often do is just comparing to the pre-IP or the surrounding tissue. So what RNAs were specifically enriched in this population versus all others that you dissected. So the early years of my lab were devoted to taking mice I'd made in Nat's lab, those that I thought were important for psychiatric disease, particularly like neuromodulatory populations and glia. So, and I had a mouse line for each cell type I was interested in. And we would try and go in and sort of pick a phenotype we thought was associated with that mouse, with that cell type, and try and understand that cell type's gene expression, and then determine whether we could use that to identify genes that regulated that phenotype. So here's an example. Here's the last of those studies we were doing in 2018 in Cell Reports, where we did the noradrenergic neurons of the locus coeruleus. This is the main producers of norepinephrine for the forebrain. And we could successfully target them and profile them. This just shows an enrichment of sort of the known genes that we thought were expressed in those noradrenergic neurons. We saw that, good depletion of glial genes. But we also identified hundreds of new genes that enriched in these cells that hadn't been previously characterized. Kind of one of the surprises that came out of that was actually this locus was very sexually dimorphic. So there was also a lot of gene expression differences between males and females, far more than other comparison cell types. And I won't go into detail because it's published there, but we picked out sort of one gene that we thought might be important for controlling these cells that was sexually differently expressed. It's a EP3 receptor, and showed that female brains specifically responded to agonists of this at a much higher level than males, and we could modulate mouse behavior in a sex-specific way. So that's the kind of work we've been doing around sort of cell types and genes within them. And when I first started this work, I was sort of, the fact that we had to purify the ribosomes from the cells was one step away from what I usually think of as gene expression, right? It was we weren't really looking at transcription per se. We were looking at the second step of translation. And then initially I kind of thought of this as a bug, that we were stuck not quite getting transcription. But over time, I came to realize that actually could be a feature instead. Is that, gene expression really is in two steps. You have transcription in the nucleus, which makes the mRNA, But then you have also this translation to make the protein in the cytoplasm. And if you think about something like a neuron that has an incredibly elaborate morphology, huge processes that can be even hundreds of microns to millimeters away from the cell body, yet you only have one source for the mRNA in the nucleus. That means that there must be substantial regulation at the level of translation to actually make the right amount of protein in the right places, to make for a functioning cell and a cell that can adapt to its environment. And especially in the advent of a lot of sort of single cell sequencing, RNA sequencing methods coming out, those can do a much faster job of identifying cell type specific expression profiles, since they're untargeted methods. But kind of a unique niche we still had was understanding how translation is regulated across different cell types in the brain. So today I'm going to talk about our efforts to think about TRAP, not just a way of doing gene expression profiling, but a way of specifically profiling translation and what we learned out of that, in terms of how translation is regulated in the nervous system, both with some surprises and some expected results. Excuse me for a moment. Sorry, guys. I should probably have muted the microphone before I did that. This is the joy. The joy and travails of having small children is, I love my kids dearly, but they are little viral incubators, and they've always been, since I had kids, I've been sicker than I ever was before having kids. Right. So just a reminder of what translation looks like, because this will come back up later, is that on a transcript, you have a coding sequence, which is the part that you actually think of as being translated in protein. It starts with the start codon and it ends with a stop codon. Translation occurs with a small ribosomal subunit starting in this 5' untranslated region, scanning until it finds something that looks like a start codon, recruiting a large subunit, translating through the coding sequence, and then the two separate at the stop codon, typically. Find recruitment to the start codon, there's often sort of a sequence-specific context, like a Kozak sequence that helps identify it. That'll come back up later. So how do you assess translation? Well, if you want to know something about the levels of translation in kind of simple systems like yeast or cell culture, there's a classic method people used to do called ribosome footprinting, where you would stall ribosomes on the transcript of cyclohexamine in a lysate, and then do this thing that's a little terrifying to molecular biologists, which is that you would then take your precious sample and digest it with RNase, thus destroying your RNA. But the key here is that the ribosome physically protects a fragment of that RNA, giving you a little 32 nucleotide or 28 to 30 nucleotide footprint of where the ribosome was physically sitting. Now, one of the secrets to modern genomics, it's kind of a tongue-in-cheek joke, is that you take a method from the 70s or 80s and add the word seq to it to create a new method. And this is what Ngoli and Weissman did in a really nice series of studies from 2009, actually till this day, but like the real several studies kind of in major journals from 2009 to 2011, kind of established and showed the power of this method of kind of adapting this to sequencing. So now you convert these little fragments into a sequencing library, and you can align that back to the mRNA and see exactly where ribosomes were at the moment of what you're translating. So we were interested in what we could learn in neuroscience using these methods, and I usually start most experimental series with something that's a bit simple and then try and build up from that. So the very first thing we did was simply try and replicate the methods that they had applied in ES cells and yeast to tissues from the CNS, to see whether or not we could kind of see the same kinds of things that they had seen. So in their, what they had seen was that there's a wide range of translational efficiencies across transcripts, and I'll come back to that later, and also a variety of different proteins being produced from the same sequences. So our first experiment was simply to take cultured cortical neurons, so we could start from cell culture as they did, and do ribosome footprinting and RNA-seq from the same cells. And it seemed like it was working, so we got the ribosome footprints were only in the coding sequence, as you'd expect, compared to the RNA-seq, which covers the whole transcript. And just as they saw, this is sort of an initial surprise in their data, there's actually quite a lot of ribosomes already on the 5' UTR, and we were able to reproduce that in these mixed neuronal glial cultures. The first thing you can look at with this kind of data is to determine whether or not there are certain transcripts that really are dynamically regulated in a way of sort of having a higher amount of translation than you'd expect by chance. You know, one of those advantages of having regulation at the level of translation is that for a given amount of transcript, you can actually make more or less protein, or the cells can make more or less protein in a regulated way. And this is defined by a measure called translation efficiency. I kind of prefer the term ribosome density, but TE, or translation efficiency, is the standard in the field. And it's simply dividing the number of ribosome counts by the number of RNA-seq counts for the same transcript. And you can end up sort of identifying transcripts that have a lot more ribosomes or a lot less ribosomes than you'd expect by chance, and thus are making more or less protein, even though at an RNA-seq level, they look more or less identical. So as they've done in ES cells, in neurons, we just first kind of look at the distribution of these values in a log scale. And you can see that, if neutral, there's about 2 ribosome footprints per transcript that we're seeing here. You can have individual transcripts that are 10-fold more or 10-fold less translated than you'd expect from the RNA-seq alone. We've translated this into the brain in a way that I don't have time to talk about today, but basically comparing trapped to nuclear RNA, you can see that that same spread occurs across cell types in vivo in the brain, and even some transcripts being up to 1,000 times more or less translated than you'd expect, given their RNA abundance in the nucleus. Going back to the in vitro studies, we next wanted to ask, to what extent is this regulated by stimuli? So we know that gene expression is strongly regulated by things like neuronal activity. And a very simple manipulation, a classic manipulation to look at that, sort of the sledgehammer first experiment, is to hit a cultured group of neurons with KCl, which depolarizes the whole culture, kind of stimulating a lot of firing activity. And compare that to unstimulated culture. This classic manipulation is known to induce transcriptional changes like induction of immediate early genes. And so we use it to also look at what happens to translation. First, looking at RNA-seq, just in terms of the numbers of transcripts, and all the gene lists are published here in this reference at the bottom if you're interested. So I'm not hiding them, they just make for boring slides if you want to know any of the individual genes. 1,200 or so genes upregulated, about 600 downregulated. This included many of the things that you would expect to see in a transcriptional study in terms of response to depolarization in neural cultures. When we looked at the ribosome foot protein data, we actually saw an even greater change. So this says that there's more things changing at the level of translation than at transcription alone. And that's mediated by a subset that are dynamically changing their translation efficiency in response to stimulation. Overall, there is pretty good correspondence to what's happening in the RNA-seq and the ribosome foot protein if you're looking across all genes in terms of their fold change. So most genes along the line would be things that are going up in both RNA level on the ribosome and off the ribosome in parallel in a concordant way. So the R squared, it fits about half. Basically half of the data is explained in what's on the ribosome is explained by changes in RNA-seq, but that leaves about half being left to translation regulation. And there's individual genes, this red or yellow one, that don't change RNA levels at all, but change specifically their loading onto ribosomes, as shown in this method. These are that same data shown as heat maps for genes upregulated, downregulated, changing TE, and a couple example genes of that kind of pattern, like that red gene here, FTH1, RNA-seq is not changing, but the amount on the ribosomes is going up, and that's reflected in change in TE. So this is basically a transcript that seems to be recruiting ribosomes in response to stimulation. We also see the opposite. This is some strange cyclin homolog, relatively unstudied. RNA-seq's not changing, but it seems to be losing ribosomes in response to stimulation. So this means that translation is quite dynamically regulated in the nervous system, and it represents this additional layer of underappreciated changes in gene expression. So the next sort of vignette I wanted to tell you guys is about astrocytes and their local translation. So I love astrocytes. My work in graduate school was on neural stem cells, which in the brain turned out to be sort of a subform of astrocyte, a radioglia. And I've always thought of them as a key neuromodulator and regulator of activity in the brain. And so when we as postdocs back in the Heinz lab were dividing the brain up into different cell types to go after, I took neuromodulatory populations like serotonin and norepinephrine producing neurons, as well as glia, like astrocytes and neurons. So the story I'm going to tell today comes from 2 students in the lab who really led this part of it, Chrissy Sakers and Rebecca Owinga, who both just defended recently and moved on. And it comes from first this idea of, kind of as I was alluding to earlier, this very complex cellular morphology that you have in neurons compared to like a lymphoblast cell, a little round cell. Here you have these long elaborate processes, yet all of your mRNA has to come from the nucleus. Um, so how do you, create proteins and get them where you need them, given this complicated cell, but mRNA only coming from one place? And Oz Stewart, particularly in the early 80s, showed by EM that you could identify what looked like ribosomes right at the base of these dendrites out in at the base of these spines and these dendrites far from the nucleus. So this suggested that there was a role for sort of a regulated local production of proteins to enable the formation of these synapses. This is interesting because it's known that synapses, to allow for learning and memory, or at least changes in synaptic strength, you need new protein synthesis. So this suggested that if you want to strengthen particular synapses, one way to do it is to locally produce the proteins that you need there to make it for a stronger synapse. One of my favorite classic experiments kind of demonstrating this phenomena came from Aaron Schumann way back, actually over 20 years ago now. It's still a very elegant experiment. So there's this form of synaptic plasticity called, this is chemically induced long-term potentiation, or LTP. So with this application of BDNF, you can see that you get an increase in the field potential over time. And what she showed in this set of experiments was that you could slice off the dendrites away from the cell body, physically removing them, and still get this increase in LTP, even though they have no access to the nucleus. So the synapse is capable of doing this independently. However, if you added an anisomycin, an inhibitor of translation, the synapses were no longer capable of doing this independently, or at least the synaptic layer was no longer capable of doing it independently, which says that some kind of local translation in that layer was required for this long-term potentiation. Right. So we wanted to be able to kind of look. So we were curious about what particular mRNAs were found at the synapse. This has been studied in a few parts of the brain before, like this, where you could physically slice away the dendrites from the cell body. But most of the brain's not organized that way. It's organized in a way that the cell bodies and dendrites are densely intermingled, so they wouldn't be amenable to sort of physically trying to separate them. And we wondered if there's a way we could adapt TRAP to this. Now, this is just another cartoon of TRAP, and a reminder that to mention that I've received royalties related to TRAP in the past. So what Rebecca set out to do was to see whether or not we could use TRAP to get specifically at these synaptic or ribosome-bound mRNAs from different cell populations of the brain, to add a layer of cell type specificity to studying local translation in neurons. And looking back at the different TRAP lines that have been made in the past, here's one for those Purkinje neurons in the cerebellum. While most of the signal is in the cytoplasm, in the ER near the nucleus, you could see when you crank the gain in the microscope that these TRAP lines that have GFP-labeled ribosomes also contributed little GFP-positive puncta out in the dendrites. So we thought that it might be possible to try and harvest the ribosome specifically from this layer. So there is an old method from the 80s, an old biochemical method from the 80s, whereby you can fractionate the brain into a synaptic fraction. So this is a synaptoneurosome was the variation that we were using. So you use a density, you gently homogenize the brain, use density centrifugation to identify a fraction that contains these fragments of cells that have a pre and post-synaptic apposition that's membrane enclosed. And it had been shown for a long time that these things were translationally competent. If you stimulated them and add radiolabeled nucleotide, they'd take it up and synthesize new protein. So it was sort of an in vitro way of looking at local translation. However, it has no particular cellular specificity in those fractions. Like I was saying, the way of modern genomics is to take a nice method from the 1980s and add the word seq to it. So we took that same method and tried to start sequencing these fractions. Furthermore, if we combine this with a TRAP line that labels specific neuronal classes, we should be able to isolate ribosomes by TRAP, specifically from this fraction that were contributed by just one type of neuron in the brain. So that would allow us to disentangle sort of a dense mesh of lots of different cell types mixed together with lots of different dendrites, and specifically enrich for those ribosomes coming from just one cell type, allowing us to ask the question of which cell types put which transcripts at on ribosomes near their synapses. So when we start experiments, I usually start with the simple ones. So we do just a pan-neuronal line in cortex. We picked this line because it when we're optimizing biochemistry, we don't want to start with noradrenergic neurons where there's only 1,200 in the brain. So we did pan-neuronal, but also in the cortex, which is one of these areas where their cells are densely intermingled, because the local translation hadn't really effectively been studied with them before. I'll just make a note. Also, we have this, people interested in astrocytes, we have this astrocyte line that labels every astrocyte in the brain with the trap construct as well. So actually, in terms of the neurons, findings, I'll refer you to Rebecca's paper. She also had another one just come out in eNeuro last month, comparing different cell types. One of the things we were intrigued by as we kind of developed and got this method working in neurons was what's going on with the astrocytes. So astrocytes are originally thought of as a support cell in the brain. They make up this broad symphony of cells that basically cover the entire brain in a matrix, physically kind of making contact with every micron of the brain, more or less. They're evenly dispersed from one another. Individually, they have a beautiful, elaborate morphology. I think this is sort of underappreciated because many of the classical stains for astrocytes, such as GFAP, would only label the large caliber processes. But if you actually fill them, you can see they have lots of fine processes. And it started us wondering whether or not they had to deal with the same challenges as neurons do in terms of having a localized synthesis of protein. And they have a variety of important functions in the brain that really makes me think of them as sort of like a distributed kind of neuromodulator. They're responsible for making sure the ion concentrations around the neurons are buffered properly. They take up neurotransmitters and may respond to them as well. They clearly provide a lot of metabolic support to their surrounding neurons, regulate blood flow, and control the surrounding circuitry. Interestingly, when you focus in on those really fine processes of the astrocyte and you look at where they are relative to neurons, there's been in this emerging concept coming out of sort of the tripartite synapse. We usually think of a synapse of having a presynaptic side and a postsynaptic side, but what Ben Barris and others pointed out is that for every synapse in the brain, at least 80 or 90% of them, you can always find an astrocyte process ensheathing that synapse, or at least making very close contact with it, this peripheral astrocyte process, or abbreviated PAP here. And those processes seem to, if you looked at the proteins they had, have the proteins available to do a lot of the specialized functions you want to see to support a synapse, such as taking up excess neurotransmitter like glutamate, recycling back glutamine to the presynaptic side, buffering calcium and potassium. And any one astrocyte is making these kinds of contacts with up to 10,000 synapses. And we wondered whether or not they might also have some kind of local translation at these peripheral processes. So Chrissy set out to test this question, and to establish this, she'd want to know, kind of establish three things. First, for local translation, you want to show, like Stewart did, that there'd actually be ribosomes and mRNA present in those processes. Second, that active translation is actually occurring, that they're not just there, but they're functioning. And then, that could all be true, but still kind of be boring, that just sort of everything is translated everything in an astrocyte. So the third thing that she wanted to see was whether or not it's actually a specific subset of mRNAs that are enriched out there. Right. So using microscopy at the light level, we could already see that while much with the neurons, well, a lot of the signal of these ribosome GFP mice in the astrocytes was in these proximal large processes. If you zoomed in on the fine processes, here's the GFAP positive large caliber process. You could also see sort of coming away in dashed lines, little puncta of GFP that suggested it was getting out into the fine processes as well. Repeating this at the EM level with an anti-GFP antibody developed with kind of a DAB aminoperoxidase reaction, Chrissy was able to actually image individual astrocyte processes, wrapping a pre- and postsynaptic neuronal component, and see that the ribosomes were indeed right out here in these appositions. So the ribosomes look like they're there, at least the GFP tag is there. Second, we wanted to see if the mRNA was out there. The problem with just staining mRNA in a brain of full of astrocytes is the astrocytes cover the entire brain. So it's a little bit like looking for flakes of snow in a snowfield. So we needed some way to image individual astrocytes. So she developed this method where we do a P2 intracranial injection of a diluted AAV9 under GFAP driving a fluorophore. And this creates sort of a sparse label of astrocytes in the brain, allowing us to image individual ones and sort of ignore their neighbors. And we'll use this same trick for a few different experiments. First, at the level of in situ hybridization for a prominent mRNA in neurons for the GLT1 transporter, we were able to show that those mRNAs also make it out into these very fine processes. So ribosomes are there and mRNAs are there. Next, we wanted to see if translation actually occurs. So she used that same prep of sparsely labeled astrocytes and then a biochemical trick for detecting new translation. And this is all published now in PNAS 2017. So we'd make an acute slice from one of these brains with sparsely labeled astrocytes, the same kind of slice you would do for electrophysiology. And we'd incubate it with pyrimycin, which is an inhibitor of translation that is a tRNA analog. So it binds to the extending nascent peptide on translating ribosomes. And the other thing, so it installs translation, but the other thing it does is that it also provides you an epitope. So if you have antibodies of pyrimycin, you can now detect that and identify where translation was occurring when you gave the drug. So doing very short pulse labeling in slices, this is what this looks like. You can see there's strong labeling in the surrounding neurons because they have, they certainly have high levels of translation, probably arguably a bit higher than the astrocyte. But if we use the GFP of the sparsely labeled astrocyte as a mask, we can also identify translation going on, specifically in the astrocytes. And this extends all the way out as far as the processes go. So this is quantified here as the amount of pyrimycin at different distances from the nucleus. And you can see that it extends, not in the nucleus much, but it extends pretty much throughout the cell body, all the way out to these fine processes. So ribosomes are there and they're translating. Are they translating anything interesting? And this is where we took the methods that we had originally developed for neurons and asked the questions, will they also work on these astrocytes? So we simply swapped in the astrocyte line here at the early step and asked whether or not maybe some of this peripheral astrocyte process was co-fractionating. And if so, could we harvest mRNA from it? And this is when I really knew we were onto something, which is that our first look at that gene list of the transcripts that were enriched in this fraction, and just kind of identifying the top 10 or 20 or so, and looking at what those genes were. Looking back at this cartoon from the review paper I highlighted earlier, for every one of these functions, we could find a gene. So here, you know, these processes are supposed to take up glutamate. Here are the two most highly expressed glutamate transporters and astrocytes that we're seeing enriched out in these processes. There's a specific potassium channel. Here, there is the two enzymes that are involved in recycling glutamate to glutamine here. So it was really kind of compelling that we might have actually discovered an astrocyte local translator. In addition, we had some things we hadn't thought about ahead of time, but that makes sense. So we have a variety of motor kinesins and myosin proteins. There has been some evidence from other labs that these processes are modal at a relatively slow rate, like across circadian time, for example. So this local translation of these proteins might be involved in kind of motility of these processes. And then some things we didn't expect, like an entire pathway for synthesizing fatty acids. I don't know if that's to make new membrane or to support neurons in some way. We haven't really followed that up. If we do GO-term enrichment, many of the categories I just talked about are significantly enriched. And there's also some really interesting secreted proteins too, things here that are known to regulate. Astrocytes play a key role in the promotion of neuronal synapse development, and some of those regulators of synapse development were things we were seeing as potentially locally translated. So it's a subset of mRNAs that are enriched there. We then did a little bit of work to try and identify what was driving that enrichment of those things. It looks like in general, the things that we were seeing enriched there had longer UTRs and coding sequences. They had a more structured UTR as well. So we think it's something in the untranslated region. This fits with what had been seen in neurons and other systems where motifs across a range of transcripts, they're found specifically in UTRs, seem to be involved in regulating localized translation. So you'll often have a motif in a UTR that's bound by a specific RNA binding protein, and those things can then interact with other enzymes to change RNA localization, stabilization, degradation, translation. They're Swiss Army knives. They can often do lots of different things. So the next thing that Chrissy did was to try and identify specific motifs that might be found more in the PAP-enriched sequences than a control set of depleted ones. One that she found was this motif that's supposed to bind the QK RNA binding protein, the QRE, and she confirmed that that's sort of enriched there. And we're doing a lot of follow-up experiments now to determine if that really is driving the localized translation and expression of these things. And I might save that for another day. But I'm trying to convince you, I hope I've convinced you at least that these astrocytes, which have been also, like neurons, have a regulated local translation for specific transcripts. And I think there's a lot of great questions that to pursue there as far as whether or not that's regulated activity. Can independent peripheral processes do that independently of one another, or is it do it astrocyte-wide? Is it required for any particular biological function? And the lab is interested in that, and anybody who's interested in studying those things, feel free to reach out and contact me. So kind of as a first step at understanding whether or not, we were very interested whether or not like local activity near a synapse might modulate local translation in an astrocyte. And we were starting to design experiments around that, but I realized that nobody had actually even looked whether or not activity in neurons in general stimulates translation in astrocytes. So starting with the bigger picture question first, we did a series of experiments to kind of get a sense of whether that could be true before we zoom in on the individual processes. So using this same system, it's nice because you can do pharmacology in these slices and do a lot of fast experiments. So Chrissy did a range of things that are thought to stimulate stimulate neuronal activity in these slices, or emulate the activity of neurons like BDNF, and then look at what happens to translation. She also did inhibition of activity through blockade by TTX or combinations of TTX or KCL. This is data from individual cells, so kind of 30-ish cells per group, showing what happens at baseline. You have a certain amount of translation in the astrocytes. As a negative control, we can block all translation and show that signal goes away. And then she showed that stimulating with things that should depolarize neurons dramatically increase the activity in astrocytes. So it looks like they're responding to that. Blocking activity of neurons, there's some basic activity, suppresses that translation in the astrocytes. And blocking while stimulating gives you sort of an intermediate phenotype, which says that some of the response to KCL in the astrocytes is due to neuronal activity, and some of it is independent. Furthermore, BDNF also looks like it stimulates translation in astrocytes, which is interesting. We did one additional set of experiments in collaboration with Yi Zhang, so we're curious whether or not neurons are required for this. So with Yi, she's got a nice culture system that can make elaborate astrocytes in culture. We repeated this series of stimuli in cultures that have no neurons now. Under those circumstances, neither TTX or KCL alone had any effect on translation in astrocytes. So it seemed like that was being routed through the presence of neurons. BDNF still had some. Confusingly, putting them both together did create a significant effect. We're still puzzling that one out. So astrocytes can have a translational response to neuronal activity. We're trying to get that down to the level of individual PAPs if we can sometime in the near future. And then kind of the last vignette I wanted to talk about, and what do I have, about 10 minutes left?
Yeah.
Yeah, I think we can do this in 10. I guess the nice thing about doing this online is the no question and answer period. So I get to use all the time just for my science. You'll have to text me or email me with questions if you guys have them. So this was led by a great postdoc in the lab, Darsan Sapkota, who was kind of also interested in new method development around TRAP. So I'm going to start by reminding you guys about alternative splicing. So this is the peptide neurotransmitter gene, and this is to remind you that the alternative splicing is sort of an evolutionary solution to creating more different kinds of proteins by using the same size genome. So if you have a set of exons that can be used to make a particular protein, one way you can make more than one protein from the same set is by having alternative splicing, where you use different sets of exons or even splice junctions with the same exon to make different transcripts from the same gene. And this allows, in this case, to make two different neuropeptides from the same gene. There's also, I think that's kind of a widely understood phenomena. And this does, on average, genes have, I don't know, some 3 to 10 different splice isoforms, but there are individual examples that have hundreds of different isoforms. What I wanted to talk about a little bit today is also that there's another layer of regulation that can happen even after splicing that we're starting to call alternative translation, just to have that parallel splicing. So, what if you have a gene that has no alternative splicing? Here's this CEBPP gene, which has only one exon. Well, there's still an ability to generate diverse different proteins out of that single transcript by changing what part of it gets translated. And this is what we're calling alternative translation. And this particular gene is a classically studied one in that regard, where there's 3 known protein isoforms, a full length, a lap, and a lip isoform. And these are mediated by, in green, different start codons. So these are 3 different methionine positions. And depending on which one gets used, you can make shorter and shorter versions of this protein. And that can have a clear functional consequences. This is from a review kind of showing the cartoon of this gene and these different isoforms, excuse me, even the sub-ice, 2 different kinds of the lap. This particular protein is a translation factor, or sorry, excuse me, a transcription factor. And it has activation domains at the N terminal and DNA binding and dimerization domains at the C terminal. And it works as a dimer typically. So when you have a full-length protein, this is an activator of transcription because it has these activation domains. When you have this truncated form, it can dimerize, but it doesn't have any activation domains. So it actually works as a dominant negative on transcription. So the exact same gene can have opposite functions depending on which protein isoform is produced from the same transcript. Another example of sort of alternative translation, one that's not changing protein sequence, is a sort of a regulatory one. And this comes from the stress-related transcription factor ATF4. This is another genome browser shot showing that here's where the protein is typically starting from this longer transcript. But there's actually a number of things that could potentially be start codons that are in the 5' UTR that are upstream of that translation, annotated translation initiation site, ATIS. And in classic studies from Sonnenberg's lab, he's actually shown that these upstream ones, under certain circumstances, can act as decoys. The scanning ribosome will bind there and initiate at these upstream open reading frames and translate these products here that don't seem to make a functional protein. Rather, they simply prevent access to the primary translational initiation site, preventing translation of this protein normally. Under certain circumstances, in this case cellular stress, a phosphorylation of an initiation factor can change the preference for these upstream sites, allowing for translation of the actual protein. So this is a mechanism by which biology can regulate translation of the main proteins. The transcript can be there waiting for some kind of signaling event to allow for translation through the mechanism of having these decoys in the 5' UTR. And this can be assessed to some extent using a variation ribosome footprinting. And this is pointed out by Angolia Weissman again. What you do is you add a particular inhibitor of translation or a class of inhibitors that stall ribosomes after initiation, but allow them to progress if they're already elongating. And if you look at ribosome footprints after this, without drug, you get ribosomes and some of these upstream sites, but then you also get ribosomes extending into the protein. If you add this drug and allow time to occur, the ribosomes that are in the protein elongate off, and those that remain are those that are sitting at initiation sites. And that allows you to see where initiation sites were occurring in the protein. And in this example, actually, this is their data from ATF4 from their paper. You can see the classic upstream ORFs that have been identified before. They were able to recapitulate now using this method. The advantage being of this method now is they could do this genome-wide because it's using transcriptomics and sort of evaluate all these at once. So using the same experimental design I alluded to earlier, with and without stimulation in cell culture, starting in culture because it's easy to get drugs in, we can look specifically for these sort of N-terminal variations using Homo Harringtonum. So both production of a normal protein at an annotated site, upstream events that might be in frame and extend an N-terminal, downstream events in frame and extend, truncate, like the example of the CBEPP, or these out-of-frame upstream events that could be making some kinds of decoys. Darsan first kind of repeated their experiments they'd done in ES cells in the brain to see if many of the same sorts of things were true, and then to try to confirm that these things actually were making protein or not. And he identified a variety of novel protein isoforms, and we just put this paper out, I think, a month ago in Cell Reports, so you can get more data there. So here's an example of some of our traces from these ribosome footprinting. And we can identify for this one transcript actually quite a number of peaks that appear to be translating, including, here's the annotated translation initiation site, but a variety of new upstream ones. One of the surprises, though I guess it was true in the ES cells, is sort of for any given transcript, on average, we're seeing at least 3 different translation and initiation sites. So this suggests that quite a lot of protein diversity could be generated by this mechanism and or these kind of decoy mechanisms. And some transcripts had up to 12. And at that point, we almost stopped counting beyond that. We were worried about our read depth being too shallow. But it's actually kind of the minority that only had one translation initiation site. So for any given transcript you're looking at, there's a lot more proteins being made or potential proteins being made than you would traditionally expect. This is just to show that many of these are in frame, but many of them are out of frame. So on average, about half are in frame and half are out. And there are also events, as Angolia saw, that aren't starting on ATGs, but rather things that are near cognates. We also saw that in our data. We confirmed a subset of these working with the STEAM lab at Harvard, who do mass spec on similar cultures. We confirmed that a subset of these are actually making proteins. So here's peptide evidence showing that for this RTN1 gene, we actually are seeing a novel sort of 20-ish amino acid extension where it's starting from this upstream start codon, changing the end terminal of the protein. Here's an example where we had a downstream initiation site and peptide that confirmed the existence of that sort of initiation at the second methionine and not the first. So we did a fair amount of bioinformatics to try and understand why particular if you have multiple translation initiation sites, why are some selected over others? The Kozak sequence, that sort of motif around the start codon, seemed to have some influence on it, with the tendency of the annotated one to have a stronger Kozak than either DTIS or UTIS in terms of how well it matches that canonical motif. And if you looked at how in general, I guess this kind of also says the more a TIS was used, the stronger the Kozak was. So there's sort of a correlation there between those things. Again, with the annotated one tending to have a stronger one. So when you were seeing an annotated one not being used, to what extent were we seeing the use of decoys, like the example of that ATF4 gene? So we did see that the stronger the Kozak sequence was around one of these potential upstream TIS sites, the less you use the annotated translation initiation site. So if they had kind of better scores, you got less usage. That seems this decoy phenomenon is kind of general and can explain, you have this negative correlation here, so it can explain some of the data. Likewise, the more of those decoys you had, the less you had translation at the canonical site, and the longer the 5' UTR, the less you had translation. You can combine all of those things together into a single model. If you add all those variables together, you can explain about 50% of the variance in the use of the annotated one by these upstream events. So we have some idea of why you might be using less or more of the annotated translation initiation site. Contrast for the downstream ones, we couldn't really explain much. We know our assumption was that maybe the ones that had downstream initiation just had very weak annotated start codons, but that turned out really not to explain much. So that's a bit of a mystery there. What's going on? The next thing we looked at is how do these things change dynamically? So are they regulated in response to activity using that same KCL stimulation paradigm? This is all of the events we could measure well, and we're showing that in response to KCL, a lot of them shifted ribosomes from the decoys onto the main TIS from what we could see, but there are individual ones that went the opposite direction, moving to upstream or downstream TIS as a response to activity. So it seems to be the first answer is that it is regulated. The second answer is that it seems to be transcript specific how it's regulated. There's not one general rule for all. And the third thing we learned is that the magnitude of this kind of looks like the magnitude of alternative splicing. So shifts in 10 to 30%, not like all or none kind of events, but probably the same kind of magnitude of biology is what you see there. Here's some examples of individual events there. So here's with stimulation, this GFAP moves from the annotated to a downstream one. With stimulation, this HSP, heat shock protein, shifts to the annotated one. Just examples of those. So the last thing Darsan looked at was, in addition to kind of these variations of the N terminal, are there variations in the C terminal? And what this had been observed in a paper done et al., also associated with the Weissman group, was that occasionally when using ribosome foot protein, you'd see this surprising effect where a subset of ribosomes would extend past the first stop codon into the 3' UTR, but stop at the second stop codon, a phenomenon called stop codon read-through, which is relatively understudied and explained. So could we see this in our data as well? So again, I'll remind you, this is sort of the ribosome footprint. So it usually ends at the stop codon. So we looked for instances where we were seeing a pileup of reads beyond that in this region between the first and second stop codon. And so MAP2 as a negative control gene doesn't have that. But here's an example of a few things we found in the cultures that did. So here's aquaporin 4 and MDH1 both had robust read-through, but only until the second stop codon. We wanted to confirm this in an independent system and to see if it was sequence dependent. So we cloned those fragments out into a reporter. So just taking this sequence between the stop codons and dropping it between two different flavors of luciferase or reporter vector, and then measuring how much we got of the enzyme activity off of this second gene, with positive controls, including things that basically were fusion proteins with no stop codons or negatives with lots of stop codons. And we can see that these sequences for aquaporin 4 and MDH1 were capable of supporting read-through on their own. So it was just something about those sequences themselves allowed the stop codon read-through to occur. So the next thing we wanted to ask is, does this occur in vivo? Or is it just a kind of weird artifact of cell culture? And is it cell type specific? And so here what Darsan did, and this is one of those times when like one slide represents 2 years, quite a lot of work, is that he figured out a way of coupling our trap method, which allowed for cell type specific capture of ribosomes, with this ribosome footprinting approach. So essentially digesting the mRNA while the ribosomes are on the beads, leaving these fragments. So now we can look at this alternative translation in vivo in the brain in a cell type specific way. We haven't done much with the N-terminal variations yet because getting the drugs into the brain at the right dose is going to take some optimization, but he did focus on the C-terminals to start with for this paper. And this is just showing for that aquaporin 4 gene, specifically from their astrocyte line. This is an IGV browser shot. I should update that. But here is the open reading frame. Here, the thing with no red interruptions until you get to the first stop codon. And then here's the second stop codon. And you can see that you have reads piling up in vivo between those two stop codons, just like in vitro, meaning that the read-through was probably occurring in vivo. So he focused for the remainder of the paper on this aquaporin 4 transcript he'd seen. It's an essential water channel, highly expressed in astrocytes. It's interesting because the knockout can ameliorate or exacerbate stroke, depending on the model. And it's a target for autoimmune disease, this neuromyelitis optica. It also seems to have an important role in potentially clearing A beta from the brain, though the mechanism by which it does that is sort of controversial at this point. It's known to be localized in a specialized process of the astrocyte called the end foot. So the end foot is this part of the astrocyte. You have these astrocyte fine processes that make contact with synapses. You also have a portion that makes contact with the blood vessels, and this provides part of the key blood-brain barrier that makes the brain sort of uniquely specialized in terms of interaction with the vascular system. And aquaporin 4 is highly, not only there, but is highly enriched there. And there's some potential sort of perivascular space that's involved in clearance, lymphatic-like clearance potentially in the brain, or normal. Some debate there at this time I won't get into. So the first thing that Darsan did was he wanted to confirm that this thing was actually making a protein and made what we thought it did. So working with people at Cell Signaling Technology, we raised an antibody to that novel C-terminal extension sequence. So if we're adding amino acids, we made an antibody that's specific for those. We call that aquaporin 4X, or extended antibody. This is compared to a pan-aquaporin 4 here in green. So the pan-aquaporin 4 antibody does label blood vessels, this kind of structure here. It also labels basically the surface of every astrocyte of some density as well. The interesting thing about this aquaporin 4X antibody is that it actually seemed to specifically localize to the blood vessel channel. So it suggested this read-through was having something to do with protein localization. We're super excited about it. We were pre-replicated, aka scooped, by another lab on it shortly after we got it. Well, you should check out their paper too. They did some really nice functional work characterizing whether or not these channels have normal flow. And they did it in rat and human. We did mouse, so slightly different, but same findings on that part. This is Darsan's quantification showing that the X form seems to be more strongly perivascular than the regular aquaporin 4 antibody, which of course is going to label both the stuff at the end feed and everything else. The other thing that Darsan went on to show was you want to know whether or not this read-through phenomenon is regulated. And so we started by looking in stroke, since it seemed to be involved in stroke. after a stroke, if you look at the peri-infarct region, all of aquaporin 4 is upregulated in the gliotic astrocytes, but the aquaporin 4X is actually upregulated less. That's quantified here. So it seems like really there's some kind of differential regulation between the two that's possible. And we looked across multiple ways of inducing gliosis, whether a genetic lesion or a physical injury, and this seemed to always be true. So there seems to be some regulation by gliosis of this read-through phenomenon, suggesting that it's biologically, the localization suggests it's functional, and then this suggests it's regulated. And we're kind of, Darsan's working on following that up now, as well as its relationship to disease. So this is kind of my take-home messages for the day that I'm flashing back to. I think the last one I was talking about, hoping to convince you, is that there's a whole layer of underappreciated regulation in terms of alternative translation and different proteins that can be made from the same exact mRNA sequence if you look for it. And we found this is just one of, I highlighted just aquaporin 4, but he found this happening in about 1% of the protein of the transcripts we could measure in the brain, and in a cell type specific manner comparing neurons and glia. So there's sort of dozens of transcripts this is happening too. And we're following up the ones we can. And then the last and most important slide is to thank the people that did this work. I think I tried to highlight throughout the people that were, the bulk of the astrocyte experiments I was talking today were Chrissy and Darsan. They collaborated with a couple of computational people in the lab that wrote some of our ribosome footprinting steps, Chengran Yang and Ali Lake. There's an undergraduate who did a lot of the work on the activity-dependent work, Rohan Kaczynski. And then this is the whole lab, who was a real pleasure to work with on all of our projects, including these. And of course, our funders who made all of this possible, and our collaborators who make our lives and our science better. So that's what I have today. Normally I would ask for questions, but I don't know if there's been some through the chats or anything else.
Yeah, there's nothing there. Like I have one or two questions because you can just
yeah, you want me to switch it back to the other
view?
Okay.
Just unscreen.
Let me get myself arranged here. There. Okay.
Okay, thank you very much. It's pretty interesting. I learned a lot from, I haven't been following this part of the whole thing much. I have one basic question, so I don't know whether it makes sense or not. Like, you have been doing these various transcription, translations, and all those things. Like, can you multiplex them?
You mean I would.
At a time you do 5, 6 of them or 10 of them, because in the disease state, what happens is some are up, some are low. If you have consider one, for example, Alzheimer's, some things go up, some things go down. Like, if you can multiplex simultaneously, hey, this form is kind of up here, this you can.
Oh yeah, so that the ribosome footprinting combined with TRAP is all transcripts at once, as long as you sequence deeply enough. So I was highlighting aquaporin 4, but we have those same measures genome wide. And so that's all in the supplement. I tend, for these papers, you tend to have the discovery phase where you write, here's all of the genes we found, and they're in this giant supplemental table. And then you have the phase of I can't work on all of them, so I've got to pick a couple, so you kind of zoom in. And so I might have zoomed in a little too fast. But yeah, with all of these methods, the local translation, the ribosome footprinting combined with TRAP, you're really getting a genome-wide view of what's going on. So you're multiplexing that way. In terms of just processivity, how many samples can I handle at a time? You want to do this kind of thing with biological replicates. I think in a given day, kind of a 4 to 6 sample number is pretty good for what you can do TRAP on comfortably and not get overwhelmed by the amount of samples that you're handling. And a good experiment, if you're comparing cell types, N of 2 or 3 is actually fine to see differences between cell types, because those are 10, 50-fold differences in a lot of these things. If you're doing knockout versus wild type, I think your N needs to be more 5 or 7 per group. And in that case, probably you're talking 2 or 3 days of experiments to get all your RNA in the freezer. That's just for the TRAP part. Some of the library prep, depending on the method, is easy or hard. It could be another week to 3 weeks, depending on what you're doing there. But that's sort of standard molecular biology, handling 12 to 20 samples at a time, or robotics if you get creative. But we haven't had the need exactly to bring that online, / the funding to do 100 TRAP experiments at once.
Yeah, this is what I was thinking is take a disease, for example, and because there are people do individual proteins. So if you can, like, since you have the whole thing in there, it's basically how do you put those things together and get 5 6 of them, because that might give you more answer on some of
Yeah.
what is happening.
Well, I mean, the other thing we're interested in doing, I think this is what you're alluding to, is like combining these different methods, right?
Yeah.
So, can we put together the trap method with the peripheral astrocyte profile, like the local translation method, with the ribosome footprinting, and get like local production of isoforms in response to activity? And so those are, you just need the right student who's willing to kind of tackle trying to harmonize those things. I think it's feasible on paper, and it would be really cool and informative to do it.
Yeah,
Yeah,
I mean,
that'd be
like,
fun.
the technology out there, I mean, like, you need the resources, and maybe
Yeah.
you can use AI to do some of the stuff, like.
Yeah, and the longer we do it, the better we get at it. The experiment that we have some more and more kind of these pieces in place to do these things too, and so you can scale up as you've been through it more times.
Thank you very much, Joe. I really appreciate your time and effort in putting it out there and coming on Science Hangout and
Yeah.
giving.
Well, thanks for the opportunity. This was fun and interesting. And if anybody has questions, feel free to email me.
I'll send all the emails out to people. Like, if they have any questions, anything they are looking for, any positions, they will contact you
Okay.
directly.
Yeah, I'm
Okay,
very
so
I'm very easy to find on the internet. So
I'm just a
okay.
mediator here. Thank you very
All right.
much.
Thank you. Bye.